With that, we're going to move right into the next speaker. And this next speaker needs no introduction to the EHE community. Dr. Rubin has been instrumental in EHE research for many years, and EHE patients worldwide appreciate his dedication to finding treatment and a cure. Dr. Rubin will be presenting on lessons learned from a genetically engineered mouse model of epithelioid hemangioendothelioma. I am so excited to learn more about this and to welcome Dr. Rubin.
Hi, everybody. So I have to say, this whole journey with EHE has been really interesting and very fun. It's so impressive to see how this group has, the gene fusions were first published back in 2011. So 9 years later, we've had the formation of all these foundations, the establishment of annual meetings for research, lots of funding for research, lots of new research, and I know it seems like it moves slowly to you guys, but from a research perspective, this is a phenomenal success story. Let's get started. And of course, my screen is frozen. Can you guys hear me? Yeah, I just rebooted my screen. Again, it keeps freezing. Maybe if I just go over to my screen freezes like every 5 minutes. All right, I think it's unfrozen now. All right, so sorry everybody, I'm having some technical difficulties here with this complex system. The reason we developed a mouse model was pretty clear to everybody that when we first started doing this research, we had a lot of problems trying to establish any kind of model for the disease. And so we took a lot of different shots on goal. We tried to do cell-based models, xenografts, but we also realized that a genetically engineered mouse model could push the field forward quite a bit if it was successful. We just knew we needed a living biological system to study EHE biology and to serve as a treatment model. And you can see from a lot of the talks today, they all come with caveats because people aren't really studying EHE. We're studying contrived systems to study the fusion or some aspect of the fusion. So we developed this model to sort of circumvent those problems. And so the initial questions that we wanted to ask when we developed the model was, is it sufficient for TAS-CAMTA1 to promote tumorigenesis at an endogenous expression level in vivo? And that was really something that wasn't clear when we first started doing this research. We believed it was the case, but in fact, it hadn't been formally demonstrated. And many people, even today, have raised, what is the importance of the fusion versus a deletion in CDKN2A or whatnot? And so we've answered that question definitively now with the mouse model. And I'll present that data here in a moment. We also really were interested in what TAS-CAMTA1 transcriptional program looked like in both human and mouse. And we wanted to develop this EHE disease-relevant tumor signature to give us some biological insights into the cancer. Okay, let me see if I can get there now. Okay, I'm on, so hopefully I'll stay connected. So this fancy diagram here is essentially how the model works. I don't know, can you guys see my arrow, or is there a pointer? Is there a pointer that I can use or no? I guess not. Okay. Well, anyway, if you look at the top of the diagram here, what we've done is we actually took TAS-CAMTA1, we made the fusion artificially, and we stuffed it into the normal WWTR1 locus. And so the reason we did that was we wanted it to be expressed off all the normal promoters and control elements and at normal endogenous levels. And that just means the levels that are normally present in a mouse. But we put it in the off configuration, so it's actually inverted. We stuffed it into one of the introns, and in an inverted orientation. And what we do is we're able to turn it on using a genetic trick, which is something called Cre recombinase, where we can flip it around and turn it on. And so as the mouse starts off its life with two essentially normal WWTR1 or TAS loci, when we inject the mouse with tamoxifen, it turns on this tumor gene, TAS, WWTR1 CAMTA1, at the normal locus, recapitulating what is seen in humans. And of course, the big question always is, what happens when you do that? So what happens is the mice start developing tumors. And interesting, we haven't really talked about this a lot today, but TAS-CAMTA1 is just absolutely specific for the presence, for the formation of epithelioid hemangioendothelioma. It's not seen in any other cancer. And that in itself is an open question, why that's the case. But in fact, and Jordan's going to present some data later on that kind of shows you can do things with other versions of oncoproteins, but they don't really recapitulate the disease in the same way that this unique TAS-CAMTA1 fusion does. And that actually points a lot of fingers at CAMTA1 as doing some pretty unique things that we still don't understand. And part of my laboratory is focused on the study of CAMTA1 and how that influences EHE. But in any case, what you can see here from this Kaplan-Meier curve is that starting about 150 days after we've injected tamoxifen, the mice start coming down with cancers. And so really what we're showing here is just percent with tumor. And the lines that go down are when mice form and have tumors. We use two different forms of Cre recombinase. One of them is actually an endothelial specific Cre. And we did that because we thought maybe we'd see other tumors with another Cre called ROSA. And ROSA is expressed off of a ubiquitously expressed gene in mice. But in fact, these mice have really, really similar phenotypes. And so it doesn't matter what Cre driver you use really here, as long as it's expressed in the right cell type. And so the median time for tumor development was 284 days in the endothelial specific CDH5 Cre versus 359 days in the ROSA Cre. So the latency was a little bit different, but it's not statistically significant. And the penetrance is quite high. 2/3 of these mice will develop tumors over this time frame. So that's a nice model when you can get tumor development at this penetrance. And that's without any other genetic changes. This is just TAS-CAMTA1 being expressed in these mice. Okay, my field is frozen again, so I'm just going to refresh. Give me one moment. Okay. Give me a moment here. Sorry. Okay. Seem to be unfrozen again. All right, and when we look to see where these tumors arise in the mice, they arise at sites that are really typical of humans. And so we saw a lot of them on peritoneal surfaces. And so as we've talked about today, pleural and peritoneal EHE is very bad. We actually see most of our tumors arise on those surfaces. They involve liver, lung, and soft tissue. And so we think that this recapitulates the disease seen in mice really, or in humans really well. I'm still frozen. Okay. Screen keeps flashing on and off. Okay. And this just shows what the tumors look like. So you can see on the left are the normal organs, and that's diaphragm, and on the upper left, in the lower left is liver, and then on the upper right is diaphragm that's involved by EHE. So all those little tumor nodules are all over the surface of the diaphragm. And then in the lower right, you can see that that liver is diffusely abnormal, and that's just full of EHE nodules. And then here's what the tumors look like. I'm not going to show a lot of tumors today, but this is a liver tumor. It's actually the first tumor we ever saw in one of these mice. And you can see that it's an epithelioid tumor with that funny myxohyalin matrix, which is so characteristic of EHE. And I've shown this actually to a number of human pathologists who always diagnose it as EHE. Here it is at higher power again. You can see the vacuole cells or the blister cells that Christina talked about this morning, but I guarantee that anybody who's looking at these slides who knows anything about human EHE pathology is going to recognize this tumor as being EHE. It's an absolute dead on, looks just like the human disease. And if you look at this slide, I'm sorry. So we actually stain it for vascular markers just to prove it's endothelial. And so you can see CD31 stains this tumor very nicely in a typical pattern we've seen in a human EHE. And other vascular markers are also positive. In fact, the tumors express ERG, they express CAMPTA1, just like human EHE. And then what I've done here is I've got a slide of a mouse EHE next to a human EHE. And I know that for those of you who aren't pathologists, I hope you can appreciate that these look really, really similar. And they're similar because they're the same tumor. One's in a mouse and one's in human. Now, to start establishing some real genetic correlates of the mouse model, we first looked into human tumors. And so we actually did deep genetic transcriptome analysis of 6 human EHEs. And we compared those to some other vascular tumors, namely Kaposi sarcoma in red, that's KS, angiosarcoma, that's the kind of teal colored one. EHE is purple. I don't know if that means anything to anybody, but for today it's purple. And then hemangioblastoma, which is a tumor that's associated with Hippel von Hippel-Lindau syndrome, is in green. And you can see here that EHE forms a nice tight cluster when you look at the transcriptomic analysis. And then if you actually look at what's being expressed and overexpressed in these different tumors, EHE is in fact enriched in YAP and TAZ targets. And so, as we might expect, this is a YAP or TAZ-driven cancer, or sort of an oncogenic version of TAZ-driven cancer. TAZ is enriched in EHE versus these other tumors. And so EHE here in this violin plot is black, and you can see CCN1, SERPINE1, TGFB2, CRIM1, AMAT-L2, and ankyrin D1, which are all targets of YAP and TAZ, are overexpressed in EHE compared with these other tumors, which we know are not driven by, or we suspect anyway, are not driven by YAP and TAZ. Okay, I think I might have missed a slide. No, okay. All right, and then using some sort of bioinformatic techniques, we actually wanted to get out what is the real gene signature of EHE. I mean, what genes really specify EHE as compared to other cancers? And so we actually used these pairwise comparisons of EHE versus angiosarcoma, EHE versus hemangioblastoma versus Kaposi sarcoma. And also we compared EHE versus liver, because a lot of the tumors that we sequenced came from liver. And so to really purify it down, we got to a 93 gene set. And all you have to realize is all these things intersect. And when they intersect, it allows us to remove a lot of the overexpressed genes, or thousands of overexpressed genes, to get down to 93 genes, which we think are absolutely key and uniquely expressed in EHE as compared with all these other tumors. Frozen. Oh, there we go. Not frozen. Okay. So, and then just the bottom line, this is just going to look like a laundry list here. This is the 93 gene EHE specific signature. And what we've done is highlighted and underlined a lot of known YAP and TAZ targets, which is no surprise. And we knew this from some of our other studies. But basically, these 96 genes here are a core signature now of EHE that we've established. And when you now go back, so we established all that using human data. And now when you go back and you look in the mouse tumors and you use that gene set and ask, is it enriched in mouse EHE? The answer is yes, unequivocally with unbelievable statistical significance. So in fact, basically saying that the human and mouse tumors not only are histologically identical and occur in the same anatomic site, but in fact are genetically identical now. And when you actually look at just overexpressed genes, this isn't the 93 gene set now, but you just look at overexpressed genes in both mouse and human EHE at a pretty kind of rough level, it's essentially 50% overlap. So you've got, 3,000 genes that are overexpressed in the mice, 2,000 in human, and 1,057 of them overlap. And you can see the statistic there is 1.78 times 10 to the minus 359. That's a really low number, which means the statistical significance is tremendous. Okay, and then just to summarize what I talked about, I kept this talk very short because I knew that the idea was to have time for discussion. So I just basically just described our EHE gem that we have developed. There's no real major differences as far as we can tell between expressing the CRE, which is the trick we use to turn on the gene, between an endothelial or Rosen-driven promoter, basically telling us that in fact it's just unbelievable how specific this gene is for EHE. I've worked on mouse models my whole career, and it's the first time I've ever had such a pure phenotype. Typically, you get a variety of other cancers which actually contaminate and cause the analysis to be a bit difficult. So we've got a dead-on perfect model here. It demonstrates that endogenous levels of TAS-CAMTA1 are sufficient to drive EHE tumorigenesis in vivo. So for those of you who challenged me over the years, thank you for your questions. But in fact, it only takes TAS-CAMTA1. You don't need anything else. You don't need some default in the immune system. You don't need other genes. You basically just need this one fusion protein expressed in the right cell type at the right time, and you get cancer. You get EHE. We've identified this EHE gene signature, which I think was really useful for the field now. We showed extensive overlap between the human and gem EHE. And again, one of the things that we always say, maybe it's almost slightly inappropriate here, is that TAS and YAP are important in cancer and disease. So, those of you who are living with EHE or have friends and relatives with EHE and the researchers in the field, TAS and YAP are ubiquitously involved in cancer. EHE is one of the best examples of how TAS and YAP can drive human cancer by co-opting the Hippo pathway. But in fact, we think that TAS and YAP are ubiquitously involved in virtually every cancer. And so what we're studying here has relevance well beyond EHE and will be very important to cancer researchers everywhere. And this is why we've been able to bring in people from the Hippo cancer field to study EHE, because they realize these are relevant models to understand the role of oncogenic TAS. And with YAP1 TFE3 hemangioendothelioma is the role of activated YAP. And so they're just great models to study these roles in cancers. Look at the next slide. I'm frozen again. Basically, I'm just showing the people in the laboratory who did the work and our funding. And of course, thanks to the EHE Foundation for funding a lot of this work over the years. But that's all I had to talk about today. I'm happy to answer questions at this point, assuming I can hear the questions. I don't know.
Well, let's give a quick test and make sure you're able to hear me.
Yes, I can hear you.
Excellent. So a handful of questions here. So when you look at mice that don't have the artificial variation that you're putting in, do mice naturally get any type of cancer that looks like EHE?
No, they don't. No, I've never seen a mouse with EHE.
This is truly unique, tied to this genetic modification.
Yep.
Interesting. Now, in terms of the models, I know with some other cancer models, they can lose the synthetic variation through the generations. have you had this line around long enough to know whether these animals can be bred and maintain this and have the progeny also contribute as good models?
Yeah, so we've been breeding the model now for a couple of years, and yeah, the fusion's retained because it's not on during embryogenesis. And so this is a model where we can turn it on during embryogenesis, and I didn't get into the details, but it actually you don't get any mice that survive embryogenesis when TAS-CAMTA1 is turned on during development. But we don't turn it on until they're adults, and so it's carried fine, and they breed normally. The alleles segregate just according to how Gregor Mendel described this, over a century ago.
All right, we've got a question here, kind of a two-part question related to how similar this model is looking to what we're seeing with people. The first question is, do you see evidence of chronic inflammation in the mice that we see in EHE human patients?
We haven't really specifically looked at inflammation, but I don't see chronic inflammation in human patients. I'm not really sure what anybody's talking about there. The EHE is a pretty non-inflamed tumor. There's inflammation's not associated with it at all. So we don't see neutrophils or histiocytes or anything typically that are prominent in EHE. It's really just cancer cells and this sort of myxohyalin stroma. So I'm not sure what that question is getting at, but they look just like human EHEs, which are not inflamed tumors.
Okay, and they had a specific biomarker question associated with it in terms of, do you see elevated C-reactive protein, if that's something you've looked for?
No, not in the mice.
So in terms of the biomarker signature, identifying the 93 genes that seem to be a unique fingerprint for EHE, do you have a feel for whether that would be a useful diagnostic that would replace some of the more complex differential technologies that are currently used to characterize EHE, or possibly an alternative to histology analysis?
So I think that some of the talk today has been a little confusing about how we diagnose EHE. And so I think Jim Chen was talking about secondary mutations and what role they might have prognostically. But to diagnose EHE, we basically look at glass slides. If it has a characteristic morphology, we will follow up with a genetic test to prove that it has a TAS-CAMTA1 fusion. The vast majority do have TAS-CAMTA1 fusions. And so that's a very simple genetic test. You can get at that in a few different ways, but really we just look at that genetic alteration. And then for the YAP-TFE3s, we would do a slightly different version of just looking for YAP-TFE3 fusion. And then some of these really super rare fusions that Christina talked about, we don't really typically test for them now, but we would in cases that look like EHE but didn't have the other fusions. And so really there's one genetic test that people use looking for TAS-CAMTA1 or YAP-TFE3 fusion. The other genetics don't play a role, in my opinion, outside of research. Because nobody's acting on them yet. It may be that secondary mutations in EHE, which is something we proposed a long time ago, do cause EHE to be more aggressive. But how you action that, I'm not really sure, outside of maybe more aggressive therapies and so forth. So we wouldn't use the 93 gene set to diagnose EHE, but it's more gives us biological insights into pathways that are activated, and things that we might want to target therapeutically, trying to understand readouts of when we're targeting therapeutically, are we, what's happening to that transcriptional program. It just tells us a lot about the biology that at the end of the day, what I'm really interested in is treatment of EHE, especially the aggressive ones that, involve pleura and peritoneum. And I think this is the kind of biology that starts getting us to really understand the disease. And when we do manipulations of the mouse model, will help us to understand whether they're working or not.
Well, and related to that point, we heard earlier today that a number of the human patients have these secondary mutations. Are you seeing anything similar to that in the mouse model tumors, and particularly as it might relate to severity of disease?
Yeah, we actually haven't looked at the DNA yet. Something that's on our board to do. my guess is we won't see those kind of changes at the DNA level because they might take a while to evolve. Mice only live for 2 years, and so you've got 2 years to get everything done from start to finish. And so we actually take a different approach. And so our approach is, we knew that CDKN2A was mutated. In fact, I don't know if Jim knows this, but we actually published the same data that Jim used in his paper about 3 years before he did as an abstract at a meeting. It was available from Foundation Medicine, and we came to the same conclusions. But in fact, CDKN2A is lost in more aggressive versions. And so what we do is we take a mouse that's got CDKN2A loss, and we breed it with our mouse model, and then ask, do you get more aggressive cancers? And so those kind of experiments are ongoing. But expecting to see them arise de novo in the mouse is asking for a lot, because there's just not that much time to let it happen.
Got it. And to the extent you're able to share, can you give an outline of kind of what your plans are in terms of what's top priority for testing these models against? Things like drugs, diet, any other sorts of interventions, things that might trigger disease. What are your sort of short-term plans for top priorities for this model?
Well, we haven't seen anything that really looks great therapeutically so far. And so while you've heard a lot of hints that things might be working, it might be interesting, that none of them are ready for prime time and doing up sort of a mouse model. We actually have been testing some proprietary drugs that come from industry. We have been doing that sort of work. The sort of things you're hearing about today are really super preliminary. People have talked about, trying to inhibit pathways that feed into YAP-TAZ, trying to kind of work around maybe downstream of YAP-TAZ. That's what our mechanism trial was about, that you'll hear about next week. From my interest, we're really interested in how to drug YAP-TAZ. And we have a very active drug program in the lab trying to figure out inhibitors of TAZ-CAMPTA1, TAZ, and YAP. And that's where I think the money's going to be in this field. If we can control the fusion, as Jim mentioned several times in his talk, we can control the disease. Everything else is it's like circumferential. It might work a little bit, but really to control this disease, we need inhibitors of YAP-TAZ, TAZ-CAMPTA1, YAP-TFE3, things that'll inhibit those proteins. And so that's where we're putting a lot of our effort. And we unfortunately don't have a compound that we've developed in the lab yet that's ready, but there's a lot of work going on in industry to develop these kind of inhibitors. And when they surface, people typically come to us and ask us to evaluate them. And so we'll be evaluating some of them in the mouse model, hopefully in the near future.
Got it. And one interesting point in your talk, you had noted that this ends up being a particularly good model for understanding TAZ-YAP more generally, which would be of interest to other cancers. Do you see that as a potential opportunity to push this model into both research and possibly trials for other cancers that might also then generate useful information for treatment or prevention of EHE?
Well, this is the way it would work. So if we have an inhibitor that we can show works in our mouse model, which we know is addicted, people talk a lot about oncogene addiction. And what oncogene addiction means is that the cancer is absolutely dependent on basically one molecule almost predominantly. That is certainly the case for EHE. It really depends on TAZ-CAMPTA1. So if in our model we show really good efficacy, in fact, then you can take that drug, we know we could put that into humans at that point and also show high efficacy. And it will really help people to get a drug FDA approved that could then be used for a variety of cancers. And you can ask then, knowing that you have a great inhibitor of TAZ or TAZ-CAMPTA1, or a lot of these drugs will actually probably inhibit both TAZ and YAP because TAZ and YAP are so similar. You can then just say, okay, this form of breast cancer, how dependent on TAZ and YAP is it, knowing that it would have a big effect in TAZ-CAMPTA1 and EHE.
Got it. So it sounds like the point there might be, correct me if I'm wrong, if you've got a good drug here and it's working here, it may attract more investment and interest from industry because it might be more generally applicable.
Yeah, and I think I try to get drug companies really interested in EHE because since it is oncogenically addicted to this activated form of TAZ, if we think it's going to work on this slightly unusual version of TAZ, then I encourage them to bring it into human trials in human patients, because it'll be the ultimate test of is it really an active drug in people. And again, if they can register it in people and get it FDA approved through treatment of EHE, which we would expect to be very responsive to any drug that's inhibiting this unusual form of PAS, then they can use it in other cancers. And so drug companies get that story and understand it, and you know, we have a lot of contacts now in industry because of that. We just haven't had a drug. It's not that we're hiding anything. We just haven't had a drug that's worked well enough in our models to then bring into people. It's a difficult target. TAS and YAP turn out to be very, very difficult targets. Yeah.
One last question. Since you've had such a long history of this and sort of been following the research, obviously in your own lab and other labs, you know, to the extent the community should be looking for places to dedicate additional resources, what in your opinion would be in the short term a valuable additional area that research funds and effort should be expended to advance the clinical story?
Well, I actually think there's quite a bit of research going on, and it's all in good places. Now, I've been really adamant since day one, and Hugh and people like, you know, people involved in the EHE Foundation, like Jane Gukovich, can tell you we've argued endlessly about the need for development of models. And so I'm really happy to see Patty now joining the group and pushing the development of models. And so for my money, if we're going to really understand this disease and the diversity of it, the more models we have, the better. And, you know, I'm really happy to see a variety of models. Jordan's going to present some cool models that he's developed in DJ Pan's lab next. But model development is absolutely critical to the development of developing biological understanding of this disease. And so the more models, the more different ways we can develop of looking at it at this point, I think that's where I want to put my priority. I don't think that there's a, you know, killer experiment that's just waiting to be done right now. You know, I mean, we're interested in the microenvironment, and I didn't get into this, but that's one of the things you can study now in the mouse. And so we're embarking on a big study to look at all of this stroma and all the things that surround the EHE cells and what the roles there might be, because we do think there's a lot of crosstalk between the tumor cells and the microenvironment. So that's, you know, an area that we've actually submitted a grant on now, and we're developing collaborative networks to really dig into that. I think that's an area where we can study now, because we have this one model, but the more models we have, the better. And I'm happy to see model development and also the biorepository of the tumors and you know, other kinds of samples that would be available from patients, I think are real important. That's the kind of stuff I think that's good to put money into right now.
Well, thank you so much for your time today. Everyone always loves hearing from you, and look forward to hearing more in the future.
Thanks very much. Sorry for the technical difficulties.